As for biochemistry, we're now starting the last third of the class. So at the very beginning, we talked about some things that are important like pKa, that kind of stuff. And then we started talking about DNA structures and DNA properties. And then in the second third of the class, we talked about proteins. Okay, we talked about amino acid characteristics. We talked about the different types of amino acids. And we also talked about how secondary structures are formed, tertiary structures are formed, some functions of proteins. Okay, in particular, we talked a lot about hemoglobin, right? Hemoglobin is just a protein that carries molecular oxygen to different parts of your body. Okay, so now this third of the class, it's gonna be more dense of information in a way because we're going to cover the structures and some functions of carbohydrates. We're going to talk about lipids, okay? Functions and structures of lipids, all right? And then we're going to get into enzyme catalytic mechanisms. We're going to get to essentially enzymes, okay? And then we'll talk about enzyme kinetics and that kind of stuff, okay? So this last third, we'll have a lot of material that we'll have to cover. All right, so for carbohydrates, a couple of things that are important. I want you guys to understand and familiarize with the nomenclatures of sugars, meaning the names of sugars and how they are named, essentially, okay, or the structures of sugar, like D sugar, L sugar, that kind of stuff. You should know the difference between structural isomers, enantiomers, diastereomers, anomers, and there's one more called epimer. We'll talk about that today. And this is very important. Typically, sugars are D sugars, okay? Remember, we have L amino acids and D sugars, okay? Just always memorize that. The most abundant natural amino acids are L amino acids, and then sugars are D sugars. And I want you guys to be able to understand sugars by their Fischer projection and Haworth projection. So two types of structures that we use to visualize sugar molecules. And then lastly, we'll show you how sugars can be derived into various other compounds. All right? Now, right off the bat, we're going to talk about some of the functions of carbohydrates. Okay? Obviously, all of our energy, actually I shouldn't say all, but most of the energy comes from the sun through solar radiation. And on Earth, planet Earth, we had a lot of CO2. Okay? And usually CO2, we refer to that as inorganic carbon. Okay, so it's not really useful for much of anything, really. It's pretty inert, so you can't do much chemistry with it. Okay, then what carbohydrates are, essentially, it's a storage of energy, okay, in the form of carbon-hydrogen bond. Okay, so essentially carbohydrates, because now you see how you have hydrogens attached to the carbon, you have energy that's stored in that bond. Okay, so what plants can do is they take solar radiation through photosynthesis and they convert these essentially energyless compound, CO2, right? CO2 has no energy like biologically, okay? And then it takes these inorganic carbon and we're going to put energy in it. Okay, and then that gives us our carbohydrate. Okay, and then our carbohydrates are then utilized by heterotrophs like us, or E. coli is a heterotroph. And then what we can do is we take out the energy stored in CH bond. Okay, so we take out the energy stored here. We use that to generate ATP, which is then used to fuel different metabolic processes in our human body, or used to fuel different activities, okay, in different animals. Okay, and then as a result, the oxygen, not the oxygen, the carbon here gets completely oxidized again to become carbon dioxide, and then you use water, sunlight, and then to reduce it back to carbohydrates. Okay? So essentially that's the carbon cycle on planet Earth. Okay? At least related to biology. Right? So carbohydrate number one function is obviously energy storage. So there are many other functions related to carbohydrates. For example, molecular recognition. Okay? You guys are familiar with blood types. You know how you have A blood, B blood, O blood, AB blood. Okay? So that's all that's referring to is different sugar antigen that's on your red blood cell. That's it. That's it. Okay? So A blood type has a particular sugar antigen. B blood type has another type of antigen. O blood type has no antigen. Okay? Right? AB type has both. Okay? So what those antigens are, they're made out of carbohydrates. Okay? And because each different sugar that you have, right, it will give it a different shape. Okay? So as a result, you can have molecular and cellular recognition because different sugars have different shapes. Okay? So their 3D structures will be different. Cell-cell adhesion or communication obviously will then be different. You have cellular protection. Okay? For example, in bacterial or plant cell walls, right? Those are made out of carbohydrates as well. Okay? So you see your trees out there. Trees are fairly strong. Okay? They're very durable material. And of course, they're so strong because their cells have very rigid cell wall. Okay? So what carbohydrates can also do for you, in addition to energy storage, it can also provide structural support. Okay? And very good structural support. You have some other stuff like cell signaling, cell adhesion, some biological lubricants, etc., etc. Okay? So carbohydrates plays many different roles in biology, right? And there are different kinds of carbohydrates. In Biochemistry 1, we will only introduce you guys to the basics of carbohydrates. If you guys are interested in carbohydrate research, I guess you can talk to me. I'll tell you more about it. All right. So to start, let's get some nomenclatures or some terms out there. Ultimately, that's the fundamental basis that you have to have in order to communicate with biochemists. Okay? So sugars. Okay? Sugars all have the general chemical formula of C2O and then parentheses N. So it will usually have equal numbers of carbon and oxygen and twice the number of hydrogen. Okay? So that's the general formula for sugars. And when we say you are one sugar by itself, we call that a monosaccharide. Okay? Mono obviously meaning one. Okay? Such as, for example, glucose. These monosaccharides are the building blocks for sugar polymers. Now, when you have two monosaccharides, for example, two glucose, you add them together, then you form a disaccharide. Di meaning two. For example, here shown is the maltose. Maltose 是那个麦芽糖, okay? Maltose. So glucose, this is one glucose, and you plus another one glucose, you have maltose. When you get to more monomers or more subunits in your saccharide chain, you generally just call it polysaccharide. Okay? Just like polypeptides, you have polysaccharides. Right? Now, a very good example is amylose. This guy is found in starch. Okay? It accounts for 30 to 40% of starch. Right? And it's a sugar or glucose polymer. It's a glucose polymer that's very, very long. Okay? And it's actually an alpha glucose, but we'll talk about alpha beta later. Okay? Now, if you take amylose, you degrade it, you can degrade that into maltose. And then maltose, you further degrade that, you'll get glucose. The key here is just monosaccharide, disaccharide, polysaccharide. Okay? Now, sometimes you're going to hear commercials saying blah, 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 blah. This oligosaccharide is really good for your health because it's a probiotic. Okay? Oligosaccharide is 寡糖. Okay? So you guys will hear a lot about that in commercials. That is because oligosaccharides are the best food for some of the beneficial microorganisms in your stomach and in your guts. Okay? Oligosaccharide usually refer to shorter polysaccharides. Okay, so usually like, I don't know, 10 subunits. Okay, we call that oligosaccharide. So again, the general chemical formula is this. Now, what you should know is that every carbon has a hydroxyl group except one carbon. All right? So for sugars, every carbon on the sugar molecule will have a hydroxyl group except one carbon. One carbon will be a carbonyl. So every carbon is hydroxyl except one, okay? Except one carbonyl, all right? Oh, yeah, by the way, N is equal to the number of carbons, typically. If N is equal to 1, you have formaldehyde. Yeah, that's not sugar, okay? It's actually very toxic. N is equal to 2, we don't usually count that as sugar as well. So usually, when N is equal to 3, we start counting it as a sugar. Okay, so you have triose, tetrose, tetraose, pentose, hexose. Okay, so sugars will always have the ending of OSE. Just like, so this is talking about essentially sugars. This could be mono, it could be di, it could be poly. Okay? Just like when you have 8, this is talking about the acid conjugate base, right? So whenever you see blah, blah, blah ose, that's telling you that this molecule is a sugar. Okay? All right? Now, for most of our concerns, we'll really only be looking at triose, tetrose, pentose, and hexose. There are 7 carbon sugars. 8 carbon, not so frequently observed, but 7 you do see sometimes. But primarily, you only see 3, 4, 5, 6. All right. Sugars. Like I said, every carbon, okay, so every carbon will have a hydroxyl group except one. One carbon will have a carbonyl. Now, the location of the carbonyl could then mean different functional group for your sugar. Why? If the carbonyl is at the terminal carbon, this guy is an aldehyde. Therefore, we call that aldose. Okay? Aldehyde sugar. So aldose. If the carbonyl is in the middle, this is then a ketone rather than aldehyde. So it's a ketone sugar, hence ketose. Ketose. Right? So for sugars, you have aldose or ketose. That's it. Okay? You're either an aldose or you're a ketose. Right? And switching between aldose and ketose is actually not very difficult. It's usually catalyzed by an enzyme, okay, spontaneous, so usually we use an enzyme to do this process. And next semester, when we learn about biochemistry 2, you will see that this is the second step in glycolysis. Okay, but we'll talk about that next semester. All right? Now, give you guys something interesting. You know, when you go out and you buy a sweet drink from the tea shop, Taiwan has a lot of those. For the most part, they use fructose. Fructose is 果糖. Okay, fructose. Okay? But if you think about it, there aren't that many fruits in this world. I mean, there are a lot, but it would be too expensive if you get fructose from fruits. Okay? So, it's not. Fructose is not from fruits. Do you guys know where you get fructose from? Some of you might know. From corn. Okay? Corn. 玉米. Right? Now, corn doesn't produce fructose. Okay, corn actually does not really produce that much fructose. Corn produces a lot of what? Starch. Okay, corn produces a lot of starch. Okay, and we just said that starch is composed of a lot of amylose, and amylose is a polymer of glucose. Okay, glucose is an aldose. Glucose is aldose, right? Then how come fructose, by the way, is a ketose, right? So it turns out, this is done in the industry. So they take a lot of corn starch, they hydrolyze it, okay, they chop it off, and then you have glucose, right? And then they add an enzyme called isomerase, and then they can change glucose to fructose. Okay, so a lot of times when you drink something sweet, if you buy a drink from, I don't know, Coca-Cola, for example, you look on the back, it'll always say high corn, blah, blah, something syrup. Okay, that's what that is. Okay, so it's a very economical way of generating sweet things for you. Okay, now you're probably wondering, like, how come I want fructose? Why not just use glucose? Because fructose is more sweet. Okay, glucose is not as sweet as fructose when it comes to our taste buds. 你听懂吗? Okay, huh? Okay. Carbon numbering system. Because, you know, we are biochemists, you know, when you're talking to each other, you need to tell them, oh, chemistry happens on carbon 3, okay? So they need to know which carbon you're talking about, okay? So for aldoses, okay, for aldose, it's super easy. Aldose, carbon number 1 is always the aldehyde carbon. It's always the aldehyde carbon. That's easy for aldose. Ketose, it's a little bit awkward because ketose, you have alcohol on top and alcohol on bottom. Okay? So for ketose, carbon number 1 starts from the end that's closest to carbonyl. Okay, so for example, if you look at this keto tetrose, the carbonyl is here, and this carbon is closest to the carbonyl, right? Because this end requires 2 carbons away, right? So obviously, number 1 is the carbon that's closest to the carbonyl, okay? So that's going to be our numbering system, all right? Now, remember a while back when we were talking about DNA, right? We had that ribose thing, you know, where we had that ring structure, and then we had a carbon here, right? Ribose. And remember that we always have the OH, and then we say that's the 3' end, this is the 5' end, okay? This is 1, 2, 3, 4, okay? Right? So that means that carbon number 1 here is the aldehyde. Okay, so we'll see how we close and we form a ring structure for sugar in just a minute. But so now you should see how we're getting the number assignments for the sugar chain. Okay, so for ribose, carbon number 1 is here. Carbon number 4 is here. That means that there was a dehydration reaction between carbon 1 and carbon 4. Okay? Just to, you know, recall your memory about DNA. All right. So we talked about different number of sugars, like different carbons in sugar. So you have triose, tetrose, pentose, hexose. We talked about numbering. So now we're going to get to chirality. Okay? So because every carbon has an OH group, okay? So for example, this carbon has an OH group, okay? And the other side is an H. And you have on the top aldehyde, at the bottom you have 3 carbons. Okay, so this carbon is attached to 4 different substituents. So if it's attached to 4 different substituents, that means that this thing is chiral. All right, so we have to talk about chirality. Now, Because there are chiral carbons, that means that there are going to be stereoisomers for the different sugars. The first type of stereoisomer we're going to talk about is the enantiomers. Okay, so you learned this back in organic chemistry, so don't worry too much about it. Enantiomers are things that are mirror images of each other. Okay, so they're a complete mirror image. Okay, so complete mirror image. So you have, for example, D-glyceraldehyde. So you have the carbon number 1 is achiral. Carbon number 2, here, this guy, OH is on the right. Carbon number 3 is also achiral because you have 2 hydrogens here. Right? So the mirror image will just be carbon number 2, but O is on the left side. Okay, so you have a complete mirror image. That's an enantiomer. Now, the most compact way to represent enantiomers is to use Fischer projection. Okay? Anybody don't remember Fischer projection? So Fischer projection is a way to look at the molecules in 3D. Now, carbon, when it has 4 things attached to it, usually it's going to adopt a tetrahedral shape, kind of. Right? So as a result, you can think of it as the shape. You can take your 2 hands, and then you make a V out of it, and then essentially they look like that. Okay, so one is 2 carbons, for example, 2 carbons coming out at you. The other 2 is moving away from you. Okay, so that's what the carbon will look like. Okay, so if you draw a Fischer projection, it's by convention that the bonds drawn horizontally are imagined as coming out at you, okay, and the bonds drawn vertically is pointing away from you. Okay, that's the convention for Fischer projection. Now, in biochemistry, again, we have D and L forms. They're the same thing as saying R and S form back in organic chemistry. Okay, so it really just depends on which particular field you're in. We refer to them differently. Okay, but for biochemistry, primarily D or L. Okay, so D sugars and L sugars, they are enantiomers. So the way that we assign whether or not this chiral center is D or L or R or S is based on the priority that we assign to the groups. Okay, now I think most of you are familiar with this, so I will not go into a very detailed explanation of how to assign chiral centers, but very briefly is that you have to make the hydrogen pointing away from you. Okay, as long as the hydrogen is pointing away from you, it's okay. And then you start to assign who's number 1, who's number 2. Right? Priority always go for the higher atomic number. Right? So in this case, oxygen is 1, and then the next thing you have carbon and carbon, but this carbon has 2 bonds to oxygen, so you can treat it as having 2 oxygen. So this one will be number 2, this one will be number 3, okay? And this thing will have a clockwise rotation, so then that's going to be R, serial chemistry, R chirality, and then in biochemistry we just call it D. Okay, so this thing will be D-glyceraldehyde. All right? And then the reverse will be L-glyceraldehyde. Okay. Fischer projection. Right? So given that glyceraldehyde has 1 chiral center right here, this is achiral because it has only 3 attachments. This is also achiral because it has 2 of the same thing. So that's also achiral. Right? So the only chiral carbon in tetros is the center one. So if you draw the stereo projection or the stereo representation using Fischer projection, that's what you get. But you can't use this to assign chirality because hydrogen is coming out at you, right? So if you want to assign chirality, you can imagine it by doing a 180-degree rotation. Then now hydrogen is away from you. Now you can assign the chirality there. So this one is going to be D-glyceraldehyde. Or you assign it and just flip it. Okay. Fischer projection gets slightly complicated when you are looking at a carbon structure that has multiple chiral centers. Now, looking at this particular figure, this is D-glucose, so it has 6 carbons. You have carbon number 1, 2, 3, 4, 5, 6, okay? Corresponding to 1, 2, 3, 4, 5, 6. Now, a common mistake that students make when looking at the Fisher projection is that, okay, so you're looking at this structure, right? You're looking at this structure. And then you say, okay, carbon number 2, OH is on the right. Carbon number 3, OH is on the left. Carbon number 4, OH is on the right. So you have OHs mostly on the right side. Okay? But if you were to look at this 3D structure, what you see is not true. Okay? Carbon number 2, let's say we look at this way, okay, from bottom up, it is on the right. But then carbon number 3 is also on the right. But this says it's on the left. Okay? So this is the place that a lot of students will get confused. Now, for Fisher projection, you always have to remember that the horizontal groups, okay, the horizontal things is always coming out at you, okay, always coming out at you, okay. So as a result, when you're looking at carbon number 2, you're looking at it from this direction. When you're looking at carbon number 3, this one, you're looking at it from the top, okay. And then when you're looking at carbon number 4, then you look at it from the bottom again. When you look at carbon number 5, then you're looking from the top. Okay, why? Because the horizontal ones, like those two, and these two, and these two, they are always coming out at you. Okay? So for Fisher projection, please always remember that this structure that you see is not the real 3D structure that you will see for the molecule. 基本上就是说，当你在看 Fisher projection 的时候，你每一个 carbon， OK， 你要记得说它的 horizontal 的2个 group 一定是 coming， 就是从板子里面出来啊，所以是往前面的，不是往后面的。所以也就是说，当你在看不同的 carbon 的时候，你就必须要记得说你在看的 horizontal group 它会出来，就往你这边出来。所以比如说 carbon number 2， 我会从这个 direction 看，但是 carbon number 3的时候，我就会从这个 direction 看。所以这些 OH groups 它在右边或者在左边会跟这个 chemical structure 它真正的形状是不一样的。So that's Fisher projection. Now, we're gonna have another type of stereoisomer. These things are called diastereomers. Okay. Now, enantiomers, they are mirror images. They are exact opposites. So every chiral center is opposite. For example, this guy is on the left. Now this guy is on the right. This guy is on the right. Then this guy is on the left. Okay, so mirror images are called enantiomers. Now, diastereomers are essentially non-mirror image stereoisomers. So for example, the two that are in yellow, what you see is that they have the same configuration for carbon 3, but different configuration for carbon number 2. Right? So those things are going to be called diastereomers. Diastereomer. Now, for sugars, okay, enantiomers are going to be labeled based on D or L. Diastereomers will have a different name. Okay? For example, here you have threose. On the other side, you have erythros. Okay? So diastereomers will have different names. Enantiomers will just have D versus L. Diastereomers have different names. Okay? Now, in this case, okay, how do you know which, like, let's say I give you a structure of sugar, okay, a sugar structure. I give you a sugar structure and ask you to tell me whether or not, whether it's a D sugar or L sugar. How do you decide? Okay, so the rule is right here. The prefix L or D or L designates the configuration about the chiral carbon furthest away from the carbonyl group. Okay, so whether or not it's D, okay, so whether it's D or L, you decide based on the carbon that's furthest away from the carbonyl. Okay, so carbonyl is right here. This guy is achiral. Okay, so we don't look at this one. So the carbon that's furthest away will be this guy. So then you look and see what is this? What's the stereochemistry of this carbon? If it's D, then you have a D sugar. If it's L, then you have an L sugar. We have another type of stereoisomer called epimer. Epimer is a diastereomer with difference at only one carbon. So in this case, D-threose and D-erythrose, they are also epimers because they differ with only one carbon. That is carbon number 2. Okay, so now we have how many different stereoisomers? We have enantiomers, diastereomers, epimer. Okay, so far we have those 3. Okay, so the numbers of diastereomers for aldose, triose. Okay, so if you have only one chiral carbon for triose, then obviously you only have 2 stereoisomers, D or L. That's it. Okay, now, so from triose, you add one more carbon in the middle, like this one. You add one more carbon on the top. So the way the figure is drawn is on top. So you have this or this. Okay. Now immediately you will have 2 diastereomers because now you have 2 chiral centers. Okay. And you will have a total of 4 stereoisomers. When you get to pentoses, you'll have 4 different diastereomers and 8 total stereoisomers. Okay. Now, for every diastereomers you have 4 different sugar names. Okay. Every diastereomer has a different name. Okay. For example, you got ribose, arabinose, xylose, lysose. Okay. Now, do you have to remember all of that? No. Okay. I'll tell you which ones that you do have to remember. Okay. So obviously, when you get to 6 carbons, then you'll have a total of 8 diastereomer, a total of 16 stereoisomer. So the total number of stereoisomer is always twice the amount of the number of diastereomers you have because of the D and L. Ketose, on the other hand, you will have 1 carbon chiral center less compared to aldose. For example, dihydroxyacetone. This guy is completely achiral. Right? This one has no chiral center. Right? So when you get to ketotetrose, then you start to have 1 chiral center right here. Okay? So then you have 2 stereoisomer and 1 diastereomer. And then when you go to 5 carbon, you have 2. When you get to 6 carbon, then you have 4. Okay? So for ketoses, you have 1 less chiral center compared to aldoses. And you should know why. Right? Because carbonyl is in the middle. The terminal carbons cannot be chiral because they're always attached to 2 hydrogen. Okay? This is a list of biologically important sugars. These are the ones that you definitely should know because in next semester when we get to biochemistry 2, just so you guys know, biochemistry 2 is very different. It's a very different class from biochemistry 1. Okay? It's actually a very, very different class. Okay? Biochemistry 2 is more like organic chemistry. Okay? We're doing a lot of chemistries, mechanisms, arrows flying there and there. I know some people will be like, shit, okay? But it's okay, it's okay. It's not too bad, not too bad, okay? I swear, biochemistry is a lot easier than organic chemistry. If you hated organic chemistry, biochem, you're going to be like, this thing's great, okay? Not really, okay? But yeah, it's a lot simpler because the number of reactions that you can have is not as many as what you have to learn in organic chem. Okay? Now, the reason why I say these sugars, at least the names, you should be familiar with. Okay? You should be familiar with the names and at least how many carbons it has, because later on when we, in Biochem 2, when we're discussing about sugars, okay, and then if you don't even know how many carbons you have, we got a problem. Okay, so that's going to be bad. So you should know, for example, these are the ones that you will see in metabolism. You have glyceraldehyde and dihydroxyacetone. Okay, so those two are found in glycolysis, which is the central metabolic pathway that is happening every second in your body. You have pentoses, for example. You have ribose. Ribose, you should know it's important because it's deoxyribonucleotide, right? Deoxyribo, okay? Ribose, it's found in DNA and RNA. Hexose, definitely. Glucose, everything you eat has glucose, okay? Unless you're eating like vegetable oil, it doesn't have glucose, but I think everything else does, okay? Everything else will have some amount of glucose in it, okay? Glucose is the major food that we digest, really. So glucose, fructose, these are some common sugars that are related to energy, okay? So we should know hexose. Haptulose, haptulose, you don't see that as much, but there's only one haptose that you will see. That is this cetohaptulose found in a metabolic cycle that we will talk about in Biochem 2. Okay? So these guys, you should be at least familiar with them, and minimum you should know like, okay, I think it's related to this, and it has this number of carbons. All right, so very quickly, sugars. When we talked about DNA, RNA, obviously you see how sugars is always in the ring structure. Okay? And then before this slide, I always draw the vertical structure, right? Okay. Now, turns out sugars, free sugars, if you dissolve in water, okay, most of the time the sugar will become a ring structure, and it's a spontaneous reaction. It will spontaneously become a ring molecule. Okay. Now, there are two types of ring structures that sugars can adopt. Okay, they're the 5-member ring and then the 6-member ring. Okay, if it's a 5-member ring sugar, we call it furanose. Okay, furanose. It's named because of the organic solvent furan. Okay, so it looks like that. Okay, without the double bonds, of course. So you have 5 carbon, it's furanose. If it's 6 carbon, it's going to be called pyranose. So furanose and pyranose. The way that this ring closure happens is by you having, okay, so the carbonyls, or at least the carbonyl carbon is a good what? Okay, so this guy is electrophile. Nucleophile, okay, so always remember, okay, so maybe you haven't went through that much mechanism back in organic chemistry, but for biochemistry, okay, nucleophile, you'll always see what, okay? Right? Nucleophile, you'll always see either lone pair electron or a minus charge. A lot of things that have negative charge or, mostly speaking, lone pair electrons, okay, those are good nucleophile because it has electrons. It wants to find a nucleus. Now, turns out as long as your hydroxyl oxygen can react with the carbonyl center through a nucleophilic attack, and then you will form a ring structure. Why is it that we only show you furanose and pyranose? You have OH here, you have OH here, you have OHs a lot. Like, how come we only form furanose and pyranose for the most part? Okay, why is it that I only show you furanose and pyranose? How come I don't say some other ring structure. 6-member ring, 5-member ring. What's so special about them? It has lower amount of strain because 5-member ring and 6-member ring produce you least amount of strain in a circle. So if you were to take, in this case, carbon number 3 and then react it with the carbonyl, you would form a 4-member ring, which is not very stable. Okay, so of course, you'll only form 5-member or 6-member. Now, depending on whichever one that is favored, you will get either a furanose or pyranose. Now, notice, okay, so here you have an oxygen, right? And it has lone pair electrons, and this oxygen lone pair electron, if it reacts with the carbonyl center, this carbon would have too many bonds. If you form a new bond between oxygen and carbon, then now this carbon would have 1, 2, 3, 4. It already has 4, and then it would have 5 bonds. Of course, that's not allowed. Carbon cannot have 5 bonds, so you have to break one bond. Now, which bond can you break? You cannot break CC bond, CH bond, because those sigma bonds are super stable. You can't break those. Now you have a CO double bond, so you can break one of the pi bonds on the CO double bond. So you will break one from here, and then the oxygen will actually grab this proton over. This oxygen is this oxygen. This oxygen is the hydroxyl oxygen there. Okay, so this O here becomes in the ring, and then the carbonyl oxygen now becomes hydroxyl. Because it becomes hydroxyl, you can have orientation. Your OH can either point down or it can point up. If it points down, we call that an alpha. If it points up, we call it beta. Now, alpha and beta are what we call enantiomers. So enantiomers only form in your sugar after the sugar becomes a ring. If the sugar is linear, you have no enantiomer, right? Because enantiomer only happens after the ring closes and the carbonyl oxygen becomes a hydroxyl group, and then it either points down or it points up. So now we got enantiomer, diastereomer, epimer, enantiomer. Okay, so you got 4 kinds of different stereoisomers. The prefix alpha is for pointing down, prefix for beta is pointing up. Now, alpha and beta can interconvert through a ring opening event, and this process is called the mutarotation. Okay, so it can open the ring, and then when it closes again, you put the oxygen on the other side. Is that okay? Right? And there's an equilibrium related to this process. And it can also be catalyzed by the enzyme as well. Okay? We'll talk about that next semester. Now, this table is in your book. It gives you the relative amount of alpha pyranose, beta pyranose, alpha furanose, and beta furanose of the different monosaccharides in water. Okay, so if you just take this sugar, you dump it in water, and you look at whether it's alpha pyranose or beta pyranose, that kind of stuff, and you'll get a distribution. Now, obviously, pyranose will be preferred, okay, if without any cell intervention. Okay, just by chemistry, pyranose, for the most part, will be preferred. Why? It's a 6-member ring. Okay, 6-member ring is still more stable than 5-member ring. Okay, back in organic chemistry, you should have remembered something about bond angles and that kind of stuff. And 6-member ring is more stable usually. Okay, whether or not it likes alpha or beta. That's interesting, right? What you see is, both are pyranose, but then how come, for example, you got 20 to 56 or 71 to 29? Why is that? Why is there a preference in the alpha or beta form? What would dictate alpha or beta? As to why you think some people want to produce alpha and some will want to produce beta, probably, if there's a selectivity we contribute that to probably some kind of a sterics or some kind of a pressure, right? Turns out a lot of times it's related to your number 2 carbon. Where does the OH point? If OH points down, then it's easier to have this guy point up, okay, to avoid steric hindrance. This you observe if you take the sugar dissolving water. So that chart you see over there, this chart is referring to water at 40 degrees. Having said that, okay, note this box, okay, while that some ring forms are less preferred, they still occur a lot in nature. For example, ribofuranose in RNA. Okay, so when we draw RNA, right, we always draw it like that. Now that's a furanose, but if you look at ribose prefer to have pyranose, right? But in reality, when it's in the cell, it wants to be in the furanose form because we have cellular functions that makes it that way. Okay, so it's not in water, obviously. Okay, I mean the solvent's water, but you have other cellular processes that will make it into furanose instead of pyranose, right? But if you only dissolve in water, of course pyranose will be preferred, but in this case, ribose will be a furanose in cells. So we're all comfortable with the different types of stereoisomers and enantiomers and why it becomes a ring structure. We're comfortable? Now, the next thing I want to show you is Haworth projection. Okay, so what is Haworth projection? This is Haworth projection. This is Haworth projection, and then this is the Fisher projection. Which one do you like more? I personally like to see Haworth projection. That's because it's just more visual. Fisher projection, however, can give you a lot of very good information. Now, here's how you would convert Fisher projection to Haworth projection. So essentially, you see that animation going already? So the functional groups on the right side of the Fisher projection will be on the bottom of the Haworth projection. Okay, so any groups that point to the right will be on the bottom of the Haworth projection. So this is, you can treat it as if it's a little boat conformation, and then it points down. So if it's on the right side of the Fisher projection, it's going to be on the bottom of the Haworth projection. And then you can see that GIF animation as you go. You see how it's on the right, and then rotate, 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 and then that guy sort of circularizes. Right? So just like that. So that's the two different projections that you have. Now, conformational isomers refers to conformational changes to the molecule in 3 dimensions. Okay. Certain conformations are favored because they're more stable. Okay. Obviously, you can check your organic chemistry textbook. For example, you have chair or boat. Obviously, chair would be more favorable than boat because everything is pointed away. Now, however, obviously with temperature at, let's say, 37 degrees, you have certain amount of kinetic energy there, so the molecule is going to rotate, rotate, and move around, so it's going to be an equilibrium between the two forms. It's just that maybe, I don't know, 70% of the time is in one form, 30% of the time is in another form, okay? So these things are referred to as conformational isomers. The configuration is not changed, but conformation can change, okay? So conformation is the same molecular structure or the same molecule, but in different, it moves, okay, in a different shape. But configuration it's a different molecule, okay? So in this case, conformational isomers are not the same as stereoisomers. So conformation is how the molecule is positioned in 3-dimensional. Configuration requires chemical changes. Okay, so if you want to change from D sugar to L sugar, you have to have chemical reactions. if you want to change from alpha enomer to beta enomer, you have to have a chemical reaction. But conformation, you want to change from boat to chair, you don't need to have a reaction. Okay, it just changes. So remember when we have 5-member ring, you can have the C2 endo or C3 endo or the C2XO, C3XO, that kind of stuff, right? All they do is they refer to the thing as the conformation, okay? Not the configuration. All right, so this is a short summary of all the things that we talked about so far. You got enantiomers, which are mirror images. They will have the same name, but different D or L prefix. And if it's D or L, they have completely opposite chirality for every carbon. If it's diastereomer, they will have different names. The example shown here is threose and erythros. Now, epimer is structures that have only one carbon different in chirality. Those are called epimer. Epimers will be important because later on in Biochem 2, you will hear a lot of things about epimerase. Okay? It's an enzyme that changes epimers, that changes the chirality of one carbon. And then you have anomers. Anomers are formed only when the sugar is closed into a ring. You can either have pointing down, it's alpha, pointing up, it's beta. Okay? And then you can have conformational isomers. This is just a list of repeat from the previous slide. Okay, so these are the ones that are really, really important. Next thing is we're going to discuss about sugar derivatives. Okay, so we talked about sugar structure, sugar nomenclature, and that kind of stuff. The next thing we're going to talk about is some kind of derivative. Right? You go from sugar, you can react it into something else. You can have sugar phosphates. Okay, you have a phosphate attached to the sugar. You can have sugar acids and lactones. For example, you take the aldehyde, so you go from an aldose, and then you oxidize it, and then you form a sugar acid. Sugar acid can dehydrate with itself. Oh, here, you can take a hydroxyl, and this is an H, take out H2O, okay? And then you form a lactone. Lactone is an internal ester. So you form an internal ester. This guy is an ester. Okay? You form an ester by reacting with itself. That's a lactone. You can have sugar alcohols. Okay? If it's an aldose or ketose, you can reduce that into alcohol, right? So, for example, here's aldehyde, and then I reduce it, and then it becomes alcohol. So, you can go from aldehyde to acid or aldehyde to alcohol. You can have amino sugars. Okay, so you swap OH with an amino group. You can have amino group on the sugar, like move free 那个维骨力的那个 glucose amine, right? 葡萄糖胺嘛. So it's glucose with an amine on it. It's this. Okay, so that's the thing that your mom will ask you to buy. Glycosides, meaning that you take sugars, you react them together, and then sometimes you can have a functionalized product on the outside. Okay? Those are you modify an organic compound with sugars. Glycosides. Okay? Cool? So these are some common sugar derivatives that we see in biochemistry. Sugar phosphates. Sugar phosphates are super important as cellular metabolites or metabolic intermediates. Okay? So these things are cellular metabolic intermediates. They don't really leave the cell. Okay? So these things we'll talk in huge, in crazy detail next semester. Okay? So when we go to Biochem 2, you're going to be like, I hate this phosphates. Okay? But for now, you can think of sugar phosphates as essentially activated form of sugar. So if you want to take sugar and do other reactions with it, you have to first activate it. Throw the phosphate on it, we use ATP. Okay? That's not super important, but essentially you should note that sugar phosphates are cellular intermediates. You have two types of sugars. We're going to talk about the first type, which is aldonic acids, these sugar acids. These sugar acids are formed by turning the aldehyde into the acid. This is the aldehyde carbon, and then you turn that into acid. Now, give you a very common example. This is the Fehling's reagent or Fehling's solution. Fehling's solution has copper in it, and essentially copper is in this case a oxidizer. It's copper 2, right? And it will react with your glucose to form gluconic acid. And this was actually used once upon a time to detect diabetes for people. Okay, so without the solution, it looks like blue, I guess. Now, after you react copper with sugar, then you form copper oxide, and then you generate a red precipitate. And it tells you about whether or not you have glucose inside of your sample. Now, gluconic acid is in equilibrium with its lactone form. Okay, so typically, when we oxidize, for example, glucopyranose, we actually just directly oxidize the alcohol into a ketone. But obviously, you see how they're the same thing. So if it's a free acid, or it's a ketone, or this is the internal ester, okay, it's the same thing. Now, a very good example of sugar lactone is vitamin C. Vitamin C is ascorbic acid, and it's exactly a sugar lactone. Now, another thing that I want you guys to pay attention to is this term called reducing sugar. So reducing sugar, what does that mean? Reducing sugar essentially is telling you that you have sugar that can reduce something. Okay? What does that mean? Okay, what does that mean? That means that, for example, if you have aldols, so this is aldols, you have this aldehyde group. Aldehyde group can pass the electron from aldehyde to become an acid. Then you would put A and it become A reduced. So that's what it means to be a reducing sugar. Fehling's reagent reacts with a reducing sugar, because if you don't have an aldehyde here, then Fehling's reagent, which is this copper 2+, will not react. Okay, so this guy looks for a reducing sugar. Okay, now, any aldoses could be reducing sugar. And when we talk about disaccharides in just a minute, you will see how some disaccharides are reducing sugars, but some are not. So reducing sugar, it's essentially saying that you have an aldehyde there. Okay. Now, we also have another thing that's called the uronic acids. They are also sugar acids, but they differ from the aldonic acid, hence the name, by the way, ALD, so aldehyde. So aldonic acid is formed by turning the aldehyde into acid, right? So only aldols can give you aldonic acid. Uronic acid, on the other hand, is different. Uronic acid oxidizes the terminal hydroxyl group. Okay, so essentially, okay, this is the gluconic acid that we saw. Uronic acid reacts the terminal OH, and then you turn that into an aldehyde first, and then you turn that into an acid. Okay, and then these things are uronic acids. All right. Now, uronic acids, for example, glucuronic acid is frequently observed as a common building block for proteoglycans. used as a precursor for vitamin C, etc. Okay, so these are uronic acid. Now, next thing we have sugar alcohols. All right, sugar alcohols, because you have an aldehyde right here. So aldehyde, if you oxidize it, it becomes acid. If you reduce it, it becomes alcohol. So if you react erythros, put electrons on it, you have erythritol. Mannose can become mannitol. Glucose can become glucitol, or generally we refer to it as sorbitol. Okay. Sugar alcohols are excellent compounds for sugar substitute. Yeah, so a lot of times if you go to nice restaurants, I think for like Western food usually, I don't think Chinese restaurants give you this thing at all. But like if you go to an American restaurant or something like that, what on the side of the table you have sugar, and then you have different brands of sugar substitutes. Okay, and one of those brands is sorbitol. Sorbitol is glucose. You take glucose, reduce it, and it becomes sorbitol. It's a sugar alcohol. Now, sugar alcohols we don't really utilize that much, okay, which is why the calorie is nearly zero, okay, because we don't use it. Another famous thing we have is xylitol. Okay, xylitol comes from xylose. Xylitol is used a lot in chewing gum because most of the bacteria or the microorganism inside of your mouth, they don't metabolize xylitol, right? So as a result, they don't use that. And then for you, it tastes sweet as well, a little bit sweet. Xylitol is not very, very sweet, but you can still taste the sweetness. And then you can chew on it. Since the bacteria, they don't degrade it, so you can inhibit bacterial growth. And then your gum will take out the bacteria for you. So xylitol is used a lot in chewing gums. And there's a brand just called xylitol. So. All right, next we have amino sugars. Amino sugars are widely found in nature, and most of the time they provide structural support, like glucosamine, right? 那个维骨力啊，葡萄糖胺。You guys have no idea. Like when I was studying United States, every year I come back to visit family, and they'll always ask me to buy like 20 bottles of glucosamine, and it's really crazy. And then every time I pass the borders in the United States, people are just like, are you taking these to sell or something? It's like, no, my mom wants it, something like that. Okay, so these things are used. Why? Okay, for example, glucosamine. Glucosamine is a component or a precursor for your knee fluid. It's for supporting your knees, okay, your knees. Now, some of these common amino sugars, you have N-acetylglucosamine, this guy. Now, this beta, D, you should already know what they refer to. Beta refers to the enantiomer. D refers to the D-enantiomer of glucose. Now, this is glucosamine, so it's no longer just glucose, right? Because it's not ose anymore, so we know it's not a sugar anymore. It's a modified sugar. In this case, gluco or glucose tells me that it's a glucose. Amine tells me that it has NH3 somewhere on it. Now, when you look at the structure, you don't see the NH3. That is because the NH3 is functionalized with acetyl. This group is called acetyl group. And N, this big N, is referred to acetyl group attached to N, to nitrogen. That's why it's called N-acetylglucosamine. You have another thing, this is muramic acid, or you have N-acetylmuramic acid. Again, N, this is acetyl, and then muramic acid, right? Things like that. A lot of these are used in providing, for example, in E. coli, you have that peptidoglycan layer. Some of these sugars are used for that layer. So these are primarily used for generating structure for cells. Right? So they just have nitrogen in them. Another thing that may be potentially useful for some I seriously don't want to say biotechnology applications. Seriously, 我真的每次跟人家讲那种生物科技，然后再跟我讲那个保健食品啊，或者是什么擦了会美白的东西啊 ，seriously， 那真的会让我觉得真的很 sad. But anyways, biotechnology. Okay, biotechnology. People are interested in cosmetics. Okay, why people are interested in cosmetics? Because cosmetics sell a lot of money. Okay, girls will spend, not girls, older ladies. Okay, especially older ladies will spend a lot of money on things that will make them look nice. Okay, one of them being hyaluronic acid. Hyaluronic acid is this thing that is found in your skin, okay, and it attracts water, so it makes your skin looks really, for the lack of a better word, bouncy. I guess that's the word. Okay, so it looks tighter. Okay, and hyaluronic acid is made out of two different sugars, okay, or sugar derivatives. One is a sugar acid. This guy is an acid. The other one is a N-acetyl glucosamine. Yes. So this one is a nitrogen. And hyaluronic acid just repeats. So it's a polymer. Okay, it's a polymer. And this is used in biotechnology or by cosmetic companies to you produce them and then mix them with cream so then you can put it on your face. Sells at a lot of money, just so you know. You also have glycosides, okay, glycosides. Glycosides are formed between two things, sugar and something that has a alcohol or something that has an alcohol. So it's the elimination of water between hydroxyl groups on sugar with another alcohol. And then you form a glycoside. And in essence, it's a way to link sugar onto something, okay? Now, for example, here you have glucopyranose and you can react it with this guy's methanol. And then you can form this methyl alpha D glucopyranose. Now, in your book, it will actually discuss two things. One is oleuben and phthalate, okay? But these two compounds are produced by plants. Okay, these are glycosides produced by plants and they're actually toxic chemicals. So in your book, if you guys have time and are interested in these two chemicals, you can actually look, there's one or two paragraphs in the book on this section discussing these two chemicals. It's actually quite interesting. Okay, yeah. This one is related to hydrogen cyanide, so it's toxic. All right, so the next thing that I want to get into is disaccharides. Right, oligosaccharides and disaccharides. So oligosaccharides and polysaccharides are polymers of monosaccharides, obviously, and the bonds are formed, okay, so here, bonds are formed between the anomeric hydroxyl group of one sugar and a hydroxyl group of another. So this is almost the same as that glycoside bond. Okay, essentially it's the same thing as a glycoside. So you have an elimination of water once you condense them together. For example, lactose. You guys know lactose, right? Lactose is the sugar that you find in milk. Now, lactose is formed by reacting galactose and glucose. Okay, so you have beta D galactose, so the OH is on top, and then you have beta D glucose. So this beta is referring to glucose OH on top. So now, the bond that you form, in this case lactose, is galactose, link it with beta glucose. Okay? So you react galactose using the beta anomer with the glucose on the fourth carbon. This is 1, 2, 3, and then 4. Okay? You react them like that, you get lactose. Okay? Now, here's some of the systems that we have to remember. Now, to distinguish the features of different disaccharides or saccharide bonds, okay, we have to know a couple of things. the two specific sugar monomers involved, so we'll say glucose, fructose, xylose, mannose, whatever. We usually just take the first three letters of that sugar and then as an abbreviation for that sugar. So you will have to tell your audience what is the sugar that you're working with. And then the next thing you have to tell your audience, what is the carbon position involved in the linkage. In this case, we have, this is 1, this is 4. So you have 1, 4. So I have 1 to 1, 1 to 2, 1 to 4, 1 to 6, etc. So you have to specify which two sugars you're using, where do they link, and the order of the link. Is it glucose first and then galactose, or galactose first then glucose? Okay, so the order matters. All right, so this is important because the second sugar will have another anomeric sugar that it can react with. Now, so the question is, let's say I have glucose, galactose, like this. I wrote it like this. I'll just write glucose, 1, 4, galactose. Is this sugar a reducing sugar? Why is it that it's a reducing sugar? Okay, because here you used to have that OH, right? This is the anomeric OH, right? If you open it, what does it become? Aldehyde. If it still has the anomeric OH, if you break that guy open, that anomeric OH becomes the aldehyde group. Okay, so it is a reducing sugar. So be confident and say, yeah, I think it is. Now, you also have to specify the configuration of the anomeric hydroxyl group of each residue. Is it A or is it alpha or beta? So for example, here I have a alpha linkage between two glucose. This guy is maltose. Maltose is a disaccharide. So you can write it a couple of different ways. So you can write essentially glucose, alpha 1, 4, glucose. That's maltose. This is different. That's crazy. So this is 1, 2, 3, 4, right? And this is 1, 2, 3, 4, right? So this is 1 to 4. Now, this guy is 1, 2, 3, 4. And then, which carbon is this guy? This is a very sick question, just so you know. This carbon is 1. How do you know it's 1? Because it's attached to the anomeric. So here is it's attached to obviously an OH, right? And then it reacted with this guy. And then it's also attached to the O here. So this guy is the anomeric carbon right there. Okay, so this is 1, 2, 3, 4. All right. So here you have alpha 1, 1. Okay, this is alpha alpha trihaloose. Next, we have glucose and fructose. You have alpha D glucose, beta D fructose, and this guy is sucrose, and that's the thing that people like to drink. It's very sweet. This is alpha 1, 2, because here you have 1, 2, 3, 4, right? That's 1. And then for ketose, remember fructose is a ketose. Where do you start numbering? You start numbering from the closest end that has the carbonyl, right? So this guy is 1, this is 2, right? Because this will be the carbonyl. All right, so this is disaccharides with alpha connections. Okay, so alpha connections will always refer to the first carbon's anomeric type. Is it alpha or beta? And as you can see, all of these are alpha, alpha, alpha. Okay, that's the alpha 1, 4. So it's referring to the left side, left sugar. Now, very, very quickly, we have, of course, this is going to be beta, right? So you have beta glucose, and then you have beta 1, and then this is 4, beta 1, 4. Okay, this thing is cellobiose, a precursor to cellulose, okay, which is used for plants a lot. You have galactose to glucose. This is lactose. So it's beta 1,4 of galactose to glucose. You have a lactose. Next, you have, oh, this one is weird, right? So here you have 1, 2, blah, blah, blah. So that's 1. What is this carbon? You have 1, 2, 3, 4, 5, 6. Okay, so this is beta 1,6. Okay, beta 1,6 is a lot of times used as a branching point in polysaccharide construction. We'll talk about that later. Okay.